Okay, adaptive immunity. We talk about the non-specific, the immune responses. Now we talk about the specific immune response. So so let's say the infection is so strong, it breaks your physical barrier. Inflammation doesn't help. Phagocytosis, they cannot eat up all the infectious agents, so the infectious agent escape the phagocytosis, and complement or interferon, they cannot help, they cannot stop the infection. Then what happens next? Next one is called adaptive immunity. Some people call it the specific immune response. Some textbooks call it humoral immune response, humoral immunity. The humor is from the humor. Humor means body fluid. Specifically, it means your blood. So what happens is that if it has something to do with the antigen and antibody reaction. Antigen, the definition is very good. I like the definition, is short and clear. Any molecule that stimulates the adaptive immune response. So antigen in here, it basically is, they are some kind of glycoprotein on the surface of the plasma membrane. Specifically, they are the glycoprotein on the plasma membrane on the pathogen. Those glycoprotein, that is like an ID card, or they have this ID card, but the ID card is not recognized by our immune system. So your immune system sees, okay, this is a fake ID card, or this is an ID card from other school or other country. Then they will start to attack the pathogen that carries this antigen. So that's why the immune system is able to tell self from non-self. And it takes about 5 to 10 days to develop the adaptive immunity. After you develop the adaptive immunity, the adaptive immunity can last for 5 to 7 years. What does it mean here? An example is getting a flu. Every year we have a flu season in the United States. If your classmate keep on coughing, sneezing right next to you, and if you did not get the flu vaccine, or if the flu vaccine and does not match the strain of the flu virus that your classmate is coughing and sneezing, then you get the infection. Meaning that you will also get the flu. Remember, what are the flu symptoms? The flu symptoms, they're basically fever, running nose, coughing, or sneezing, all these. All these, they are the non-specific responses. Usually, how long does your flu last? If you are healthy or lucky, it may last 5 to 10 days. After 5 to 10 days, you will be so-called recover. But actually, it's not really recover. What happens is that after 5 to 7 days, then your adaptive immunity activated, meaning that your body starts to secrete antibody to specifically kill that strain of the flu virus. In this case, when your body starts to make antibody against the flu virus, then your body your body will stop the non-specific immune responses like fever, sneezing, coughing, running nose. So that's why it takes 5 to 10 days to fully activate it. Similar to the vaccine, after you get the flu vaccine, if you read the information sheet, what does it tell you? It tells you that it takes 5 to 10 days for the flu vaccine to work. Why? Because it takes 5 to 10 days for your body to start the adaptive immunity. Yeah. So it's the same idea. We'll go into this in more detail in the next few slides. And then adaptive immune response involves B cell and T cells. They have the antigen receptor. The antigen receptor, they can bind to the specific antigen expressed on the pathogen. And then once they bind to the pathogen, then it will activate the B cell and T cell to work. And then they have one type of receptor. And density diversity is needed for many possible antigens because we have many different strains of the pathogen. So we need to produce many different types of B cell and T cells in order to get all the possible antigen. So what does it mean? So when B cell and T cell, they are able to bind to the specific antigen, then they will be activated. Activated meaning that they will start to divide, form a lot of clone cell. So all these cells will have identical antigen receptor that bind to that specific antigen. You multiply a lot so that only the cell that has the specific receptor will be cloned and divide, so that they will all recognize the same type of pathogen with the same type of antigen. Then they can start to attack that pathogen. So you may ask, how can we generate the antigen receptor on the B cell and T cell? Because we have many different types of pathogen. Well, so here it says that the generation of antigen receptor is a random genetic process, and because it is a random genetic process, some of the receptors, they actually can react with the self-antigen, the MHC1 protein. This will be bad because if our immune system works against our own body, then you have autoimmune diseases. For example, rheumatoid arthritis. Rheumatoid arthritis is your immune system attacking the cartilage in your joint. And then theoretically, if the antigen receptor made by the B cell and T cell, by mistake, they can recognize your self-antigen, then what happens? They will be destroyed by apoptosis early in the development. This is in theory. But if your body fails to destroy this kind of lymphocyte so that the lymphocytes survive and start to attack your own body tissues, then we will talk at the end of today's lecture. We will talk about some examples of the autoimmune diseases. So again, B cell, they can be activated in lymph nodes or spleen, and the B cell receptor bind to the antigen. So I told you that B cell is made and mature in the bone marrow, yes. But after they mature in the bone marrow, they still need to be activated, because activated B cell, it will become something called a plasma cell. Activated B cell, or we call it plasma cell are the cell that will start to make antibody. And antibody is the protein that will cause agglutination of the pathogen. And then cytokines secreted from the helper T cell. So for the helper T cell, when the antigen receptor on the helper T cell can bind to the pathogen, then you will activate the helper T cell. Helper T cell will secrete cytokine, something what we call interleukin. Interleukin 2, specifically interleukin 2. interleukin-2 is a signaling molecule to tell a B cell to divide. It is to clone themselves. So B cell is called antibody-mediated immunity because B cell will become plasma cell. And then plasma cell will start to secrete antibody. So I told you the activated B cell will become plasma cell. They will start to make antibodies to fight the infection, the pathogen. But the activation of B cell to become plasma cell, you need the cytokines from the helper T cell or the B cell itself, the antigen receptor, bind directly to the pathogen so that you will activate the B cell to become plasma cell to secrete antibody. So two ways, either getting the cytokine interleukin-2 from helper T cell or B cell directly binds to the pathogen in order to be activated to become a plasma cell and secrete antibody. Some of the activated B cell, they will not become plasma cell, they will become memory cell. Memory cell, what is it for? They are for long-term immunity. This is the kind of immunity we talk a lot every day. What it means is that if the same antigen, meaning the same pathogen, the same strain, or the same pathogen that express the same antigen, as soon as the pathogen expresses the same antigen, then the memory B cell will divide and quickly become plasma cell. So that instead of going through the non-specific immune responses like fever, running nose, coughing, sneezing, you don't need 5 to 7 days to trigger the adaptive responses because you already have the memory cell. The memory cell does not require 5 to 7 days. The reaction is almost immediate. The memory cells can become plasma cell and can start to secrete antibody to fight the infection. That's the main reason why vaccine works, because flu vaccine, for example, flu vaccine, they are either weakened virus or just the viral RNA. So when we get the flu shot, we don't get the non-specific immune response. But after 5 to 7 days, when your body make the memory cell, then you will be fine. Yeah, because if you get the same string of infection, the memory cell can immediately become plasma cell to secrete antibody. So this is the detection of what happened. You have this antigen, and then B cell receptor here, B cell receptors. You have different type of B cell receptors because and only this type of B cell receptor can bind to this type of antigen. So after this B cell bind this antigen, then this B cell will be activated to become, they will clone themselves and then they will become plasma cell to secrete antibody. So you see that, okay, antigen binds to the B cell, or the B cell receives the cytokine from the T cell. That means the T cell recognizes the antigen or the pathogen, foreign invader. Then it secretes the cytokine, interleukin-2, to tell the B cell, okay, we have the foreign invader, start making antibody. So what happens is that it will go through a clonal expansion, meaning that the B cell, after the B cell being activated either by binding directly to the antigen or receiving the interleukin-2, the cytokine from T cell, then they will start to clone themselves. So all the B cells, they will multiply themselves. It's like by mitosis. It is literally mitosis. Then they will start to clone themselves. All these B cells, they will have the information that they need to start making the antibody. So some of the B cell will become plasma cell and to start to secrete the antibody. Some of the B cell will become memory B cell. They will go to sleep, they will rest, and wait for will be like a reserve to get ready for future infection. And then after B cell finish their work, they will go for apoptosis. They will be so-called destroyed. So this is basically what I just told you. So the antigen will bind to the antigen receptor of B cell or T cell. Then B cell or T cell will divide, forming a lot of clones. And then only B cell with the B cell receptor shape that feed the antigen will undergo clonal expansion. And then most of them will become plasma cells, some of them become memory cells for future use.